Okay, so this is going to be part 3 of chapter 14's lecture, you guys. So we were talking about probability right when we ended. So let's start with these little example problems here. Mostly we're going to use our multiplication rule. So if you look here, what's the probability of getting homozygous recessive for whatever this P trait is, and then homozygous recessive for this Y trait, and then this hetero for the R trait. And you can see you got 1/4, like so if you were to do a Punnett square, the probability of getting the PP, the little p, actually let me do this for you. So these are monohybrid crosses. So big P, little p crossed with big P, little p. I only get little p's over here. Now, right away you can see that it's important to really deviate your capitals from your lowercase. So you can see it was 1/4 chance of getting that genotype. And then it's going to be, let's see here, why do they have half for that genotype? And then we got, so are they doing different crosses every time? They give us the, I don't know if they had given us a problem. Separately. So we got, cool. Okay, so then, guess depending on the crosses, so then if we had a certain cross where it was 50-50 chance of getting a little one, and it would be if we had, let's switch to Y's. See how we're going to get 50-50 now of the little guy? Okay, so that's a chance there. And then if we were going to get halves, again, that's the same cross for a hetero. So they're just trying to show you, though, that each cross is an individual thing to be considered. And then you can figure out the probability of getting these 3 different genotypes in one organism this way. So that first one is simply 1/4 times 1/2 times another half. And you can see it for the other ones. Okay, and then they have the addition one here. So then you can do what's the chance of at least 2 recessive traits. And then you would do your addition there. Okay, so then let's get to inheritance patterns that are more complex. So we have the relationships between genotypes. Now, I got a lot of vocabulary I'm throwing out here, and some of us might be feeling a little bit lost with vocabulary. So kind of spend some more time and just try and practice and grasp it and use it, and then it'll become easier. So how I remember genotype is it's the actual genes. So those letters represent the actual genes. So big Y, little y is a genotype. Okay. Phenotype is what it actually looks like. So let's say the yellow is, the Y is a yellow pea pod, or a yellow pea, actually, the seed, actually, is the phenotype. So then many heritable characters are not determined only by genes with 2 alleles. Remember, Mendel was trying to experiment and learn things and mess around, so he instinctively picked things that he noticed were only 2 different options. They were either yellow or they were green, for example. But it's not that simple. However, the basic principles of segregation and independent assortment apply even to more complex patterns. Now, these 2 things, if those words are making you stumble, segregation, independent assortment, you've got to go back to meiosis. Okay, segregation is simply when the 2 homologous chromosomes separate into the gametes. So you got a paternal chromosome and a maternal chromosome, and they're going to segregate from each other, and they're going to get into a gamete, right? That's segregation. Independent assortment happens when the homologous chromosomes do what? What do homologous chromosomes do with each other during meiosis? Yeah, they do crossing over. So now they're mixing up paternal genes with maternal genes. So like right now in your bodies, you guys have gametes sitting there hoping to be spread onto the next generation. Don't do that, because that's the last thing you want. No, I shouldn't say the last thing you want. But that would definitely be a problem for you. Okay, so you still have these gametes that are hoping and wanting, and they're sitting in there. And each of your gametes either has mom's chromosome or dad's chromosome. Now, mom's chromosome, same thing as dad, but let me just deal with mom's chromosome, was sitting in her body, right, wanting to go on, and it did crossing over between her paternal or her mom and dad's chromosome, right? So they did crossing over. So say you got grandpa's chromosome in you. It's not all grandpa's because it did what? It crossed over, right, with so then your grandpa's parents were crossing over in your grandpa's body. Okay? And that's the law of independent assortment, saying that genes are going to get into the next generation randomly. Even if they're carried on the same chromosome, they have opportunity to mix that chromosome up with the homolog. Now, I know it depends on you how well you've read this chapter. Again, some of you might be feeling like really lost. Okay, so go back and visit that chapter. Or gosh forbid, can you come in and talk to Mrs. Lonely Coop and get some help? Okay. Okay, so inheritance by characters, when we say that, we're talking about the actual phenotype. By a single gene may deviate from simple Mendelian patterns. So this is when this happens. The alleles are not completely, this is when they're not. May deviate from Mendelian genetics. Do you see that? So these are different things, different ways of getting in different patterns. So when alleles are not completely dominant. So there are cases, and this is with human height, right? Being taller is not dominant over being shorter. You're a mixture, okay, with that one. So it's not dominant or recessive. A gene can have more than 2 alleles. This happens with fur coats sometimes. So maybe the cat can be orange or black or white or even brown. Eye color has a lot of different options. When a gene produces multiple phenotypes, and that's what I also just talked about. Sometimes we got blending. So maybe then we had a black gene and a white gene for cat fur, and then when they're both expressed, right, maybe it's a gray color, or maybe it's a spotted cat. Okay, and that goes over both of those. And here's an example. Now, I really want you to pay attention to Punnett squares, because that's what you figuring it out it is. Okay, so this one is incomplete dominance, and you could see when the heterozygous is formed, it's an intermediate. But look at how we symbolize it. You guys look up here. It's not just one letter. We're going to say the color, so we're going to go with a C, and then we're going to say that allele is saying be red, this allele is saying be white, and when I write it like that, I know it's not dominant, it's incomplete dominance. And now we have a blending or an intermediate. So to get red, you have to have what type of genotype? Homozygous, right? Homozygous with the big R, okay? And again, notice there's no middle because there's not a recessive. So that white is capitalized. Okay, let me go. Where's here? So the last thing, it says, for any character, dominant recessiveness relationship of alleles depend on the level at which we examine the phenotype, what we're actually getting to see or measure, because sometimes we can't. Okay, what about diseases? Because this is when genetics is the most important to our understanding. So Tay-Sachs disease is a fatal dysfunctional enzyme caused by an accumulation of lipids in the brain. Lipids are what? Fats. Okay? So at the organism level, so we're talking to the human being, right? The allele is recessive. At the biochemical level, the phenotype, which is, if the enzyme is going to really break down the fat, is incomplete dominance, because both things could be occurring. At the molecular level, the alleles are co-dominant, where they're both working. Okay, so dominant alleles are not necessarily more common in populations than recessive. For example, one baby out of 400 in the United States is born with extra fingers or toes. 1 in 400. So we've had a good amount of people who are walking around campus right now, right, that were born with extra fingers or toes. Now nowadays, what do they probably do when the baby's first born? Yep, they just correct it right then and there. And sometimes they don't, depending on what the parents want. They'll fix fingers more commonly than fix toes, right? Some people don't. It's a decision. The allele for this unusual trait is dominant. The recessive allele is far more prevalent than the population dominant allele. So most genes exist in the population in more than one form. So then we get to blood types. So blood types here, look how they're going to represent blood types, because this is confusing. This is a capital I and this is a lowercase, because not all people even have these alleles. So this says that this allele is coding for an A-named blood protein. This one is for the B. This person's allele doesn't have either, so we say it's a little I. And a little bit more detail on it. The enzyme that encodes for the A allele adds what we named an A carbohydrate to the red blood cells cell membrane. So they have this A carbohydrate attached to a protein. Whereas the enzyme encoded for the B adds a B carbohydrate, and again, the I, nothing. They have 0. Now, in common conversation, we call that O-type blood. O, right? So they don't make A or B. The O in reality was meant to be a 0, but that didn't stick, and humans named it O. Okay? So gives you an idea here. Now think back of cellular communication. Remember our shapes and how cells can communicate and recognize each other? And this is the case here. If you give a person that has A-type blood, B-type blood, a transfusion, is that a problem? Yeah, the body thinks it's a foreign cell, which it is, but it's not going to harm us. And then the immune system starts attacking these blood cells that are supposed to help the person because of that cell-to-cell communication. Okay. Most genes have multiple phenotypic effects. This is called pleiotropy. So alleles are responsible for multiple symptoms. This is an example of cystic fibrosis and sickle cell anemia. So sickle cell is caused by one gene. It's actually one amino acid mutation. It's a substitution. We'll get to that a little bit later. But that allele causes the hemoglobin protein to be misshaped. And then it causes all these other types of phenotypes because of its change in shape. So when you think of the characteristics of, oh yeah, this individual has sickle cell, when she has a sickle cell attack, the bloods can clot, so she can get blood clots. She could get blockage in the arteries. She can just have problems with breathing and like out of air feelings. So see how we have several different phenotypes from one type of genotype. Okay, epistasis. This is when a gene on one locus, that's location. So on one gene on one chromosome alters the phenotypic expression of a gene on a second. So these genes can affect a different gene. Some of these genes are really good. They're regulatory genes. For one that is kind of cool to study is mice and many other mammals have a coat color that depends on two genes. One gene determines the pigment color, while the B for black and the little b for brown. Another gene with allele big C for color and little c for no color determines whether the pigment will be deposited in the hair. So if you have little c, even though you're saying be black in a different spot, you don't get to express that. And that pigment is never made, which is a protein. So here's an example here. So to get the phenotype, you need to consider both genes. Did you see that? And a little bit on ratios, because we're going to get to ratios here. So simply counting up for phenotypes, if you count them up, we have 9 black, 3 brown, and 4. So your genotype in this case is a 9 to 3 to 4. Now I want you to think about this. I'll just grab one randomly. This little b right here, where did it come from? This little bee that got in that rat right there, or mouse, where did it come from? What? I didn't see it, hear you. Say it again. Oh, okay, the parent rat. Yes. When, right, the eggs were made, it depends which one it could have, yeah. The little bee came from the egg. Okay. Beautiful. Notice too, real quick, that because this is a gamete, this is a gamete, this is a gamete, you only have one of each. So when you're building these Punnett squares, sometimes they get confusing. So this is haploid, and this is diploid, right? Because now we have fertilization. So in this diagram, we are representing meiosis, and that came from here. Big B, little b, big C, little c came from right here. And it's foil. Have you guys noticed that? How do I figure out all the combinations? Let's do that real quick. So on the top, big B, big C, little c. So just do foil to get all your options. So we'll say the big one is given away with this big one. And we'll say this big one is given away with the little one. In a slightly different order than I did, does it matter? No, not as long as you move through all of them. Little one with the big one. There's that one. Little one with the little one. And there's that one. I got all my possible combinations then that were made by meiosis. Okay, there's also quantitative. What does that what does that word remind you of? Quantitative. Quantity, you guys. Quantitative. What's it remind you of? Numbers. Okay, so quantitative characters are those that vary in the population along a continuum. That also is the case with height. Right? We go up in different types of height. Okay, so quantitative variation usually indicates polygenetic inheritance, an additive effect of 2 or more genes to get all those different variations. This is also, you can call it continuous. Skin color in humans also, right? We can line up, we can go like across the, what is that, a monochromatic, who are my art people? Is that what that's called? Some sort of scale there where we go from white all the way to black? Is that right? So again, now look at this here. So we have three genes, right? That contribute to how dark our skin is. And these genes are making the pigment melanin. I think I'm getting close to doing a stop real quick. Yeah.